My dear students, as we have seen into the previous figure that due to a particular phenomena, the level inside the thistle funnel is rises up to h, the density of the solution is rho, and g is acceleration due to gravity. Therefore, we can write pi is equal to h rho g, where pi is the pressure due to which the level of solvent or solution rises in the funnel. The flow of solvent from pure solvent to the solution. If solvent and solution are separated by semi-permeable membrane is observed, this phenomena is called osmosis. If we put dry grapes in a pure water, the water flows in the grapes. Let us understand the same with one particular figure. Here you can see a figure in which we have placed a dry grape inside the pure water. This pure water does not contain any sugar, and that is why we can say there is zero concentration of sugar in this particular solution. We have another thing present in the water, which is a grape, and this grape has some of the sugar present inside it and a very small quantity of water, and that is why we consider it as a concentrated solution of sugar inside the grape. What happens? From pure water, the water molecule goes inside the grape, and thus the grape swells up and the volume of grape is increased. Here, the cell wall of the grape behaves as a semi-permeable membrane. Now we look at the second experiment. Here in the second experiment, as you can see here in this figure, we have taken a juicy green grape which is having a juice of sugar. So it is a solution of sugar inside the grape, but we put this grape into the more concentrated solution of sugar, or we can say that the saturated solution of sugar. So outside the grape the concentration of sugar is more, and inside the grape the concentration of sugar is less. Here in this case, the water molecules or the solvent molecules moves outside the sugar, and the wall of the sugar or the wall of the cell of the grape cell behave like a semi-permeable membrane. When a green juicy grape is placed in the concentrated or saturated solution of sugar, from grape water, that is the solvent, flows towards the saturated or concentrated solution. Here in grape there is a sugar solution, but outside the grape concentration of the solution is more, or we have this concentrated solution or saturated solution. Thus in both the above cases means in this case or in this case, in both the above cases we notice that, what do we notice? We notice that solvent flows from less concentration to more concentration. Here you can see that here we have the more concentration outside and less concentration inside. That is why the solvent flows from less concentration to more concentration. Here we have less concentration outside and more concentration inside. So from less concentration to more concentration the solvent flows. That is what we have noticed here, that in all or in both the cases solvent flows from less concentration to the more concentration. When two solutions with different concentration are separated by semi-permeable membrane, solvent flows from less concentration solution or zero concentration solution in case of pure solvent to more concentrated solution, how, like, to what time this solvent flows? Until the concentration of both the solution becomes equal. This phenomena is called osmosis. So this is how, when we observe all this, we can understand, we can call this phenomena as an osmosis. Now we can understand why raw mango shrivels in brine. Now we can easily understand that raw mango has more concentration and or less concentration, and brine has more concentration. So from less concentration to more concentration, solvent flows and mango shrivels. Wilted flower revive in the fresh water. Fresh water, zero concentration. Wilted flower, more concentration. That is why the water flows from fresh water to the flower and flower revive. Blood cell collapse when suspended in saline water. So in saline water there is more concentration. In blood cell there is less concentration. From less concentration to more concentration, the solvent flows and blood cell collapse. So in all the above cases, water flows from less concentration to more concentration due to the phenomena called osmosis. How can we stop the flow of solvent? Now we want to stop this flow of solvent. How can we do it? To understand the phenomena better, let us take two vessels. We will take two vessels or two beakers and put semi-permeable membrane, that is SPM, between them. One vessel we fill with solution, that is more concentration, and the another vessel we will fill with solvent which is having the zero concentration or less concentration. So here, my dear students, we have the figure. You can see here that one vessel is filled which is having the solution, so it is having the black dots as well as the red dots, and it has both solute as well as solvent. Same way, we have another vessel in which only the solvent is filled, and that is why you have only the red dots. Both these solutions or both these liquids are separated by semi-permeable membrane. What is the speciality of semi-permeable membrane? Semi-permeable membrane will allow only the flow of solvent to the solution, or from the solution to the solvent, but only the red color particles, that is the particles of solvent, can flow. The particles or black particles, that are the particles of solute, they cannot flow through the semi permeable membrane. Now what happens, as this is the semi permeable membrane, we will notice that the flow of particles will be there from the solvent to the solution, from the less concentration to the high concentration, and this particular phenomena is known as osmosis. We know that outside there is one atmospheric pressure which we call it P atmosphere, or one atmospheric pressure is written as a P atmosphere. So both these vessels have automatically P atmospheric pressure present on them. Now from outside, here on the vessels of solution, we some or the other way start increasing the pressure. When we start or when we start increasing the pressure, we will increase the pressure up to the level where the flow of solvent from solvent vessel to the solution vessel just stop. Remember, my dear students, we will increase the pressure to that level, up to the level when the flow of solvent from vessel number 2 or from this vessel stop going into this vessel. The moment the flow stops, we will stop increasing the pressure. So now we have the pressure outside this vessel is equal to P atmosphere plus pi. This pi is, my dear student, the osmotic pressure, which is equal to the pressure exerted on solvent so that the solvent flows from less concentration to the high concentration. This is how, my dear student, we can stop the flow of solvent from lower concentration to the higher concentration. The flow of solvent from solvent to solution stop. Thus, we can say that the osmotic pressure is the excess pressure that must be applied to a solution to just stop the flow of solvent to solution, or to prevent the phenomena known as osmosis. So, this is how we can define the osmotic pressure. Now, if we apply more pressure, then P plus pi, P atmospheric pressure plus pi, that is your the osmotic pressure, to the solution side. Here in the solution side, we have initially applied the pressure P atmosphere plus pi. Now suppose, apart from this pressure, we also add some more pressure to this. So this is what it is written over here, that instead of only the pressure P atmosphere plus pi, let us say we now apply the pressure P atmosphere plus pi plus P1. Then the solvent flows from solution to the solvent. This phenomena is called reverse osmosis, my dear student. So now if we apply more pressure P1 to this side, then the flow of solvent starts from moving solution to the solvent, and this particular phenomena is known as the reverse osmosis. Let us understand reverse osmosis, my dear students, with one of the example. Reverse osmosis is used in the desalination of sea water. Let us understand, my dear student, how the desalination of sea water takes place. Here you can see that we have a semi-permeable membrane. Remember, my dear student, that in desalination of water, the semi-permeable membrane used is the film of cellulose acetate. In both the side of the cellulose acetate semi-permeable membrane, left side we have fresh water, which is potable water. Right side we have sea water. It is also known as the saline water or salt water or brine. This particular solution is concentrated solution or having more concentration. This solution is pure solvent or having zero concentration. From here, using this piston, we can apply pressure. So we apply pressure, and here what pressure do we apply? We apply the pressure of P atmosphere plus pi plus P1. So initially, if we do not apply this pressure, what happens? From fresh water, the solvent will flow towards the sea water. But due to applying the pressure more than the osmotic pressure, that is P atmosphere plus pi plus P1, what happens? The flow of solvent or the flow of water from sea water to the fresh water takes place, and only the water particles are allowed to move by the semi-permeable membrane, and thus from the sea water we got only the fresh water. This fresh water is simply collected from the water outlet, and that is how the sea water is converted into fresh water, or it is converted into potable water. So this is what it is, the figure that represents the schematic setup for the process of desalination of seawater. My dear student, many of the countries where there is a scarcity of water, they use these methods of reverse osmosis to meet their needs of pure water, especially the Arabian countries or the countries which are having more and more deserts with them. Now, my dear students, we want to understand few more things. Let us read this so that we can understand what is next we have to look at. Osmotic pressure is generated when two solutions have different concentration. As we have discussed, my dear students, that osmosis takes place only when the two solutions have different concentration and they are separated by the semi-permeable membrane. When two solutions have same concentration, there is no osmotic pressure. It is quite obvious that when two solutions have same concentration, the osmosis stops or osmotic pressure does not exist anymore. Thus, we can say osmotic pressure pi depends upon the concentration, that is C. At the same time, osmotic pressure is a colligative property. That is why osmotic pressure also depends upon the total number of particles of solute present in the solution, irrespective of the nature of the solute. So now what we can say, that pi proportional to the C